Thank you so much, Ole. This was, I think, a terrific introduction to all our presentation related to stem cells this morning. As you said, we agreed to have now a presentation of the IMI projects using stem cells. Then we will have the coffee break and we will take all the questions during the panel discussion, during which we will also have at the end a presentation by Ruth Markkanen. So that's the program. And to introduce our IMI projects on stem cells, I will ask Fatia to join me. Fatia is our scientific officer at IMI, who is indeed in charge of, I would say, our stem cell portfolio with 2 major projects, but also she's also looking at the use of stem cells in other projects. So, Fatia, perhaps you will be kind enough to introduce our 4 speakers.
Yes, thank you very much, Michel, for giving me this opportunity. So we'll begin with the first project, which is StemBANCC, and I will ask Zameel Cader to come to the floor. So Zameel is a consultant neurologist at the John Radcliffe Hospital and director of the Oxford Headache Center. He is a senior clinical scientist at Nuffield Department of Clinical Neuroscience and a principal investigator at the Weather Institute of Molecular Medicines. So his research program is focused mainly on understanding the disease process in migraine using cell biology, model organism, and human imaging studies. So his work has led to the first identified gene linked to a common form of migraine, and his group is now working at MRC technology to bring more effective pain treatment arising from this finding to clinic. And finally, he's also, and this is in this behalf that he is present today, the managing entity of one of our flagship projects, which is IMI StemBANCC, aiming at developing stem cells from patients as a new platform for drug discovery. Thank you, Zameel Cader.
Great. Thank you very much, and thank you for inviting me to speak today. So I'm going to tell you about the StemBANCC program, and as Fatia says, I'm the managing entity, whatever that means. And I'm very privileged to have Martin Graf as the coordinator of the program. And I have to say he's a very fantastic coordinator, and I'm also very privileged to be working with a great consortium, I think, and we've really come together as a team over the last year. And I'm just going to sort of highlight some of the achievements that we've made. So why was a program like StemBANCC formed? And why is it important? And I think it really arose from what's been apparent for a little while, that traditional models of drug discovery is failing. So that despite increasing investment, there've really not been a corresponding increase in the number of new medicines. So that's obviously led to quite significant changes in the way in which pharma operates, but perhaps has also questioned that basic paradigm itself. And as a neurologist, I'm particularly alarmed by the rising burden of neurological disorders and neurodegenerative disorders. For example, look at Alzheimer's disease, perhaps one of the biggest, scariest problems of our time. A huge number of people affected and a huge cost, and it's only going to get worse. And yet, we don't have a single disease-modifying therapy for Alzheimer's or indeed for any other neurodegenerative disorder. So what's going on? What's the problem? And we've heard already very nicely from Professor Isaacson some of the reasons why drugs are failing when they get into clinic. And one of the reasons is that it's because we identify targets mostly, for example, from genetic studies that provide the linkage to a particular disease with a particular gene or a particular protein, a target, and we then model that in cellular systems and model that in animals. And we develop assays focused on that particular target. And we make those assays fantastic. They work really well. And we identify compounds, and that discovery of the compounds works. It's robust, it's scalable, and industry like it. And of course, we then test it within that setup. And we test it, and it works in the animal model, and so we have proof of concept. And all the time, we're quite far removed, apart from that original linkage, from the human system. So initially, we have drug success, but then when we start to translate it into the human system, the drug doesn't work. And it doesn't work because we haven't captured enough the complexity of the human system in that first assay. So we get unpredicted toxicity, and we don't get the efficacy that we hoped that we'd be able to see. So this is one of the major reasons why drugs are not translating through. So what we'd really like to do is to try to bring that human system much earlier in the drug discovery process. So that's essentially what the StemBANCC project is setting out to do. And it's really about providing a platform and trying to bring and catalyze the paradigm shift in drug discovery. And so what StemBANCC is aiming at doing is to developing both the tools and the know-how to enable that. And we're focusing upon some of the biggest disorders of our time. So we're looking at neurodegenerative diseases. These are hard diseases to develop therapies for. We're looking at neuropsychiatric disorders and neurodysfunctional disorders like pain and migraine, from which pharma have really been pulling out because it's been so difficult. And we're looking at diabetes, a global epidemic. And of course, we're also trying to address toxicology. Can we be better at predicting toxic responses? So we're going to be establishing a biorepository of 500 subjects across these range of diseases and generating stem cell lines, pluripotent stem cell lines from those patients. We'll then develop and optimize the differentiation protocol so that we can develop these assays, these models, diseases in a dish that we can then use to study both the disease process as well as develop the drug screening protocols. Okay. The consortium is composed of 10 pharma companies and 23 academic institutions. We started in autumn of 2012, and we have 55 million euros over 5 years. I mentioned that the project coordinator is Martin Graff at Roche, and University of Oxford is the managing entity. We've established work packages to take us from the initial recruitment all the way to giving exemplars of how a drug screening assay at the high throughput level should constitute. So work packages around the clinical recruitment, with workflows around the reprogramming, and then the molecular phenotyping as well as the cellular phenotyping. And then finally, taking those phenotypic discoveries that are occurring in academic labs and in industry and converting them into something that's amenable for high throughput screening. Assays that are based, for example, on 96-well, 384-well plates. And that can be done on millions of compounds that are in compound libraries. So what have we achieved over this last year? And I have to say this year has really whizzed by. Okay, this year and a half now. But we've built, I think, a really effective project coordination team with a steering committee and really strong lines of communication across the work packages, across the management hierarchy. We've had strong public engagement because it's important that the public is aware that stem cells have multitude of applications, and perhaps one of the nearer term achievable goals is in the use for drug screening. And so we had a very successful launch and worldwide media coverage. We now have a StemBANCC website, which some of you may have seen, and I think it's a very good-looking website, and it's effective for communicating what we're doing, and it's also effective at communicating between consortium members. And that's, again, I think, really important, that we have a shared resource for the consortium to exchange ideas, because that's what the Innovative Medicines Initiative is all about. It's about partnership between the various stakeholders. And so we've got involved patient groups, we've got involved academia, we've got involved the ethicists, and we've got involved industry. And they're all talking to each other, working collectively in a pre-competitive space, and which is fantastic because we're all working towards a common goal. rather boring in some ways, but actually absolutely essential in order to ensure that what we are going to be achieving is actually going to change things. And that is, we're setting about making sure that the standard operating procedures are in place and the workflows are really well defined. And so that really will bring about resources that have the appropriate and needed level of quality assurance, so that when it comes to the time that we need to distribute these resources, then international researchers can reliably say, these are cell lines that are useful. We know that these are robust and that they will have the characteristics that we desire. We've established a good ethics governance framework and the mechanism by which both consortium members and then the international community can access these cell lines. And we'll be making these as freely available as possible because we want these cell lines to be used across the board. And we have multi-center, multinational ethics, which was really quite a challenge for us when we started setting this up, but we've achieved it and we're very proud of that achievement. And we've really got cracking now with the patient recruitment, which is obviously what's feeding into the ultimate biorepository. And very importantly, that clinical recruitment is absolutely essential because it's the old adage of garbage in, garbage out. You've got to make sure that what you're putting in has got the absolute top quality clinical characterization, so that researchers who are using the cell lines know what they're dealing with. So we are doing that in Oxford. A large number of patients are coming from clinical centres in Oxford with disease experts. Parkinson's disease specifically is coming from Lübeck, some autism patients and schizophrenia patients from London, and migraine patients from Copenhagen. So that's really underway and working well. We are now also progressing very well with our reprogramming. So already 60 subjects reprogrammings are in progress or have completed, and we're focusing initially on perhaps the more tractable disorders, which is the monogenetic forms of disease. We're also trying to ensure that whatever expertise we're developing is transferred from one partner to another. So we're holding workshops and seminars, and we've also started to develop standardized differentiation protocols. And we've particularly have achieved standardized protocols, for example, with nociceptive neurons, cortical glutamatergic neurons, dopaminergic neurons, and podocyte differentiation protocols. And these are some example pictures of the kind of cell cultures that we're achieving. And of course, these are things that are being achieved, in fact, by a number of groups now across the world. But I think what's important for us is that what we're achieving is reproducible. So we can see these type of pictures not just in one lab, but in multiple labs. And again, that's really important as well for achieving what we, I think, are really ultimately setting out to achieve, which is a resource and a platform that will be accessible to all and will really, I think, make a big difference in shifting that drug discovery paradigm. Thank you very much.